Thank you, Jesse, very much. And thank you also, Beat, for the invitation to give this presentation here in this wonderful conference. Is this? Yeah. So I modified a little bit the title of my presentation, and I added a section on advanced characterization for critical quality parameters. So this will be more or less the following step of development after what Danny presented on finding new concepts. Here I show you some of our machinery, how we develop, manufacture, and upscale nanoparticles which have certain selected properties. This will focus on our known technology platform of RNA lipoplex nanoparticles, which we form from cationic liposomes and RNA by a self-assembly process. We get stable colloids, which are intravenously injectable, and we can engineer them to obtain selective targeting RNA expression in certain organs. Here you see two formulations which have been developed from that. One is targeting spleen, one is targeting lung, and this has been done by optimizing the formulations with respect to the physicochemical aspects. So I will come to the aspects later on. Once you have discovered and obtained such a formulation, you have to make it as a pharmaceutical product, you have to make it under GMP, you have to make it controllable, and you have to define specifications. So there's lots of parameters you have to define in order to come from a very promising scientific concept to a product which you can apply to patients, ideally beginning with phase 1, and which you then can develop further on in order to maintain the quality, in order to enable upscaling from a phase 1 stage to a phase 3 stage up to the market. So there are all these parameters that I mentioned here have to be controlled, and they have to be kind of understood, and that's an enormous space of parameters, and you end up what we designed here as kind of a spider web. All potentially product and process parameters have to be varied, and the ideal conditions have to be identified. And of course, the ultimate readout is always biology. So you want to get the ideal signal here with using luciferase coding RNA as a reporter gene again into mice. And so you want to optimize your product towards the optimized effect in vivo. But however, the option for doing biological experiments, particularly in vivo experiments, is limited. And so you have to compromise and to find out concepts which are kind of generally applicable. So here you may try to derive some general rules of selectivity profiles, and in this example we have just plotted the spleen versus the lung and all other signals, and we could identify an optimal range where we get the spleen selectivity which we need for our cancer vaccine products. But still this is rather coarse, and so ideally you would need to make much more experiments to identify the suitable products. You can do more on that. This is from just a recently published paper where we compared also very limited parameters, just having a positively and a negatively charged product, where we assembled the nanoparticles from two types of liposomes, both having one single cationic lipid, and the helper lipid was varied. It was either selected as DOPE, a phospholipid, or cholesterol. We assembled positively and negatively charged particles from them and investigated them in vivo. And here you can see we were able to identify specific effects of the type of helper lipid inside the formulations, where as we found different profiles of intensities in the selective organs. Here you see this in a more quantitative manner. To the left, the negatively charged particles, the positively charged ones, we always find the same principal targeting priority, but here this composition is way better for targeting the spleen, while at the positive range, this cholesterol formulation is better for targeting the lung. However, these were now two conditions, and this is not sufficient. So you need much more information, much more data. And here, I have one more data point where we revealed the internal distribution in the target organ. This is the lungs, where we could also derive details in the microscopic distribution of the expression for different formulations. However, these are, as I said, time-consuming experiments which you cannot do for 100 different compositions. And therefore, you have to go back to physical chemistry and generate some underlying data. That's a data set with typical assay which is used in pharmaceutical quality control. That's dynamic light scattering, where you measure something like particle size. And again, this comparison of particles with negative and positive charge excess demonstrate that in the negative range and the positive range, particles are small and colloidally stable, and the intermediate range, they aggregate, the particle size diverges. This, of course, is still a relatively blunt tool in order to define quality, because dynamic light scattering is not so much a quantitative direct measure. Therefore, I am showing you now two further approaches to identify, to investigate a little bit more on the internal molecular organization of such products, which then help to get a generic description. This is results from binding studies, where, very simply spoken, we took a beaker of RNA and added in small doses cationic liposomes to them. And we measured different parameters, like unbound RNA by separating the particles from the free RNA dissolved in the bulk phase. This is the red data points. And what you can see is if you add more and more liposomes, the fraction of unbound RNA decreases virtually linearly, indicating that apparently products of a very well-defined stoichiometry are formed. The second data set, the blue curves, show RNA which is accessible by fluorescent dye, which is intercalating to the RNA. And here, interestingly, we found that the fraction is a little bit higher, and this is also following a certain rule, as the fraction of unbound RNA increases monotonously with the fraction of particles bound. And so you can derive a model from that, indicating that particles are formed which are constant in stoichiometry, which have a constant surface layer of accessible RNA, which points toward a model where in this whole range of particle formation, you get a similar type of product, which is very important for product control under GMP. While here in the range of positive charge excess, we expect a particle which has positive surface charge, which then also has different targeting characteristics. To investigate this further on, one method we frequently use is small angle X-ray scattering. Here you see an example from another study where we investigated fundamental aspects. In principle, X-ray scattering gives you information on structure down to the angstrom and sub-angstrom scale. Here you see, under certain conditions, these kind of Bragg peaks, which give you an information on the lamellar spacing, let's say the repeat unit inside the particles. Here you have unstructured curves which indicate this is just one single particle which scatters, from which then you can derive the internal structure. So there's in principle lots of information inserted here, which in that case enabled us to construct a model of how the RNA is inserted into lipid bilayers, and we could quantitatively calculate the composition of this phase. Now, for the next minutes, I would like to show you an example where it's an even simpler approach, where we just looked at one single peak from this positive and negatively charged particles which we assemble. And again, we varied simply the charge ratio starting from an excess of RNA and going to an excess of liposomes. And we analyzed this single Bragg peak where we look at the peak position. This gives us the information on the d-spacing, so the repeat distance which is there. We look at the peak width, which gives us information on the total number of stacks in one organized construct. And we analyze the peak area, which gives us an information on the amount of product which is formed, which is present in this particular structure. And analyzing all of this data, you can get now lots of information in the context. And starting maybe with the peak area, remember the graph 2 minutes before, when the fraction of free RNA linearly decreased. In here, this peak area in the negative regime continuously increases. So it appears that we form one defined structure of particles in the whole range of negative excess, which helps us to define manufacturing parameters. In the same range, the d-spacing, the particle, the distance between the layers is virtually constant. If you go to the positive regime, the d-spacing switches and you come up with a different organization where the spacing is significantly different. So analyzing these curves, we can now identify regimes here where either a parameter changes monotonously or is constant, which gives us the space where we can manufacture our product, and the same is true for the positive excess. And such information is kind of an important basis for defining process and product conditions, and which helps us then to come up with specifications for our pharmaceutical products for further development. And as time is running, I just would like to thank you for your attention. I'm happy to take questions, and here you see some people who contributed to that: Ferdia Bates and Sarah Noguera, who did the X-rays, Jana Bacic, Jenny, and Jens, who worked on the binding assays, and this is all more people who contributed to this project. Thank you very much.
Heinrich. Thank you very much. He deserves a prize because we finished before the first one to finish before the 12 minutes. Thank you. Questions? We open. Yeah, go ahead, please.
I like your approach of combining the animal model with the physical chemistry. However, one problem with the stability of lipoplexes is when you inject them into the animal is the interaction with blood plasma because of the falling apart due to interaction with the oppositely charged proteins. Have you considered looking into that as well?
Yes, of course we do that. This was just, let's say, one example which is pretty much straightforward, which you can present in a few minutes. And what we did is, first of all, for the physical chemistry, we looked at other aspects, and we do look also, of course, on the situation in serum. So we study serum interaction, we study protein corona, we study as well effects of RNA release on interacting with serum molecules. So this is a bit more extensive, and I like this data set because it's so concise and you can demonstrate certain aspects with few data sets. But in fact, you are right, we look at all of that. Of course, it takes time to collect all the data, but in principle, this is what we are heading for, to translate then the situation on manufacturing in vitro to the biological situation, and in principle the assays I showed you are as well applicable to a large extent for that situation.
Thank you.
Go ahead, Dan.
Thank you. I have a question regarding binding assays that you use for determination of position of RNA. Is it accessible? Is it not accessible?
Can you say it again? I didn't get it.
Binding assays that you use to see is
yes.
RNA on the surface, or it is not?
No, it's just, as I said, simply spoken, you take a beaker, add liposomes to them, and then one assay we just centrifuge, and then the particles precipitate. We measure the remaining RNA in the supernatant. Or we use agarose gel assays where
She asked, where is the RNA located in the particle?
It's our model is, it doesn't go back. Yeah, here. No, sorry.
Yeah,
Do you think that
that's
is inserted inside the bilayers?
Yes, but you said that you found that some RNA is on the surface, and I always have trouble to see where my RNA is. So that would be nice to see how you determine that.
Okay, but if you look at this one,
Before.
you have RNA inside and outside. Imagine a particle of a certain size, 100 nanometers. Then you can calculate the volume, the bulk volume, and the surface, right? And this is always apparently in a constant fraction with our system. So the remaining RNA, the accessible RNA, corresponds to a surface layer, and the fact that the fraction of free RNA is relatively constant with respect to the amount of added RNA means that we always form the same type of particles. So they have a similar size, and the surface fraction is always the same. And this helps us to define quality parameters, because in our case, the surface fraction of RNA is decisive for targeting. And with that, we can prove that we can expect, at least from this physicochemical data set, that the same targeting properties are present in the particles we form under these conditions.
Thank you very much. We have to go to the next one. Thank you. And